patients suffering from various diseases, and to find the genetic variant that lead to this, that may be causing the disease. And we also try to we also use statistics to find susceptibility to certain disease from clients, from hospitals, individuals, and population. And so before I get into the presentation, I want to talk to you a little bit about my journey as a scientist. I started my journey as a scientist as a computer scientist. I studied computer science in undergrad. I then received a master's degree in medical informatics. And at the time, I worked as a medical, after that, I worked as a medical informatics, analyzing electronic medical records to improve healthcare. And after that, I decided to do my PhD. And so after 5 years later, I received a doctorate in bioinformatics and system biology and biomedical informatics. So and after my doctorate degree, I spent some time at the La Jolla Institute for Immunology, where I study uh, 3D genomics, and this is the conformation of the DNA. And we basically analyze this data to find genetic variants that influence genetic conformation, genetic conformation, and can lead to diseases. And so after that, I started working at Ambry Genetics a few months ago as a bioinformatics scientist. Um, and so today, I'm going to talk to you about systems. Um, can So now you have you can participate in this question by answering A, B, C, or D, or E in the comment in your comment. So who knows what a system is? You have a few options here: a collection of parts, a set of rules and arrangement of things, or a group of related things that work toward a common goal. A system is a group of elements or components that are organized for a common purpose, or is it a method or plan of classification or arrangement for a goal, or all the above? I'll give you a few seconds to think about it, and you can answer. Yeah, I see C. That's right. Yeah. Um, E. I see a lot of E. Good. Pretty smart too. Yeah, so you're right. If you answer any of these, it's true. All of the above is the right answer. Um, so a system can be defined as the group of interacting or interrelated elements that are according to a set of rules to form a unified whole. It's a set of things working together as part of a mechanism or interacting network. Um, so if I were to ask you, give me an example of system, um, you can write down on the comment section again, an example of system. I'll give you a few seconds for that as well. Yes, The government, yes, that's really good. Your body, yes. The respiratory system, perfect. Yeah. Policing system, yes. Society, informed, yeah. The nervous system, environment, yeah. It's the system. I see some really, yes, you can talk.
Um, the reason I think systems are important because systems offer definition and they offer parameters. To say that you want to do something within the scientific field or any scientific field, you need to like apply the scientific method, and you need to have a system to be the vehicle to carry to carry you to your goal. But in order to know that, you first have to define your goal, then you'll understand what the parts are, and then when you understand the parts, you can get a framework, and then that framework will allow you to systematically approach, like the word systematic, like step by step, part by part, to know how you're going to reach your goals. So you from a system, you get like definition, but also it aids your research, because as you reach for something, then you'll be like, oh, I actually need the following to make my framework work, or to make my framework viable or like relevant to my desired goal. And maybe in research as well, your goal might change based on how you research your system, and then it gives like your ability to develop different systems that can be more unique or more applicable for a particular problem or crisis or an area of research that you're focused on.
Yes. Thank you for that, Zara. That is pretty good. You basically summer up our presentation today. Um, we really do care about systems because when you think of things as systems, you have the ability to learn more about it and understand it better. So, um, thank you for that. We have in the comment, I see a lot of example of systems. And to go to the basics, if you think about things around you, uh, living things are systems, even non-living things can be systems. And so, if you, oh, sorry about that. Yeah, I gave an example here. A plant is a system, a truck is a system, a cell is a system. Yeah. So I want you to think about an example of a system and think about the parts of your system. And when you think about the part of your system, I want you to think about how they all work together. Uh, Not only how they work together, but how each part works and what it contributes to the whole system. And at the end of and by thinking that way, you can better define your system and know what the function of your system is. And um, and so, as an example of a system, somebody said our body, the human body, that is a good example. Um, so if you think of a person as a system, you have many parts: the head, the neck, the arms and chest, the legs, the feet, and every single part of that system has a function and contributes to that system. In the human body, we have multiple systems. So while the human body is a system, it is also composed of many systems. We have the digestive system, the respiratory system, and some of that you gave it in the comments, which is really good. And so each part of the system has one or more roles or functions to support the system. And the reason why we care about the system, as Zeta said, is because not only do we understand the system better, we understand each part better, and we can if you think about headaches, for example, your brain, your head is divided in different sections, and depending on where the ache is coming from, you can decide whether you have a migraine or headache. Um, and if you have a migraine, if you have a headache, for example, you can look at how the brain functions, and based on your environment and other aspects that may influence that system, which is the brain, you can find out or speculate about the causes of the headache. If you're a heavy coffee drinker and that day you did not drink your coffee, then you know why you have headache. That's because you skipped your coffee. If you didn't sleep, for example, you will know. You can speculate about the causes of your headache, and you know that because you can dissect the brain in different sections, and by localizing the the ache, you can speculate on the causes of the headache. The system can also be very simple. I talked about the brain, which is a very complex system. So we have simple system and also complex system. When you have your Lego pieces or the construction pieces, when they're lying around on the floor, you don't know, you cannot do anything with one part. And so if you put them together properly, you can obtain your little toy here. You can obtain your little toy here. And so each part of this toy is contributing to form this toy, but as a single piece, you um, it can be also difficult to find out what that piece does. And and so until you have the instructions to build it, you will not know. So we have simple systems and complex systems at this point. And um, like, and to summarize this, and repeat what I said, we care about systems because it helps us understand the whole, know the parts of the system, understand what each part contributes to the system. And by knowing all of this, we can easily make changes to the system, and know what the impact of those changes might be. We can diagnose the system and repair when the system has a problem. To think of it in a more in the realm of biology, we can think about a system to better understand the system. Um, in biology, we have organisms, right? A human is an organism. We have, in this example, we have a horse. And the second example, we have a plant. And you can think about if you go from the molecular level, in this case, you have the DNA which carries the information uh, that basically define or um, give characteristics to the organism. And this information is carried by a cell. And as you can see, a cell itself is a system because it doesn't only have the DNA. The DNA is actually inside the nucleus of the cell, and this is the nucleus of the cell. And you can see that the cell as a system also has many other parts apart from the nucleus. We have the mitochondria uh, and other elements that I'm not going to go deep into. And when cells get together, a collection of cells will form tissues. And in this case, you have a collection of cells which makes the tissue. In the plant, you have the same thing. You have the cell that makes the tissue. And the collection of tissues will work together to form an organ. And with the organ, you have an organ system. A collection of organs will give you help the organism function better. Um, and so while I gave you this example for the horse and the plant, I want to talk about the molecular element, like I said, the DNA. So I said that the DNA carries the information that that basically characterize that the organism. And this is an example of that. And to walk you through the human system from DNA, DNA are basically your DNA basically are the main carrier of the information, like I said, and they, sorry, so they form a chromosome. Sorry, but okay. So you have your DNA sequences that basically form genes, and though the genes are carriers of are actually the information that will be converted into protein. We'll talk about that later. And the collection of genes, the collection of genes form what we call a chromosome. And the chromosome, as you see in this example, lives in the nucleus of the cell. And so, like I said, in this case, you can see that the cell as a system, carries the information of the organism, and the chromosome lives in the nucleus of the cell. I want you to remember that. And we have 23 pairs of chromosomes, which form what we call the genome. So you hear about genetics, we have the genes, and then you hear about genomics, that's the genome. And the genome is the collection of all 23 pairs of chromosomes. And so depending, we also have multiple types of cells, and the genetic information in the DNA define what type of cell you may have. And so the stem cell is the cell that can mature into any kind of cell. And these are the different types of cells that you have, that you may have. You have the blood cells. I'm sure you've heard about that. White blood cells, red blood cells. You have the epithelial cells, immune cells, sex cells, and others. You also have fat cells and other type of cells. So genetic information within the nucleus help inform the type of cells. And like I showed you earlier, the collection of these cells will form what we call a tissue. And in this example, you can see the connective tissue, epithelial tissue, muscle tissue, nervous tissue. And so the collection of these tissue working together will form what we have, what we call organs. And in this figure, I'm showing you different organs in your body. You have the heart, the intestine, the brain, the lungs, the glands, the urinary system. I'm sure, and I hope that everybody has seen something like this somewhere in their science class. And those, and so these collection of organ systems will form what we call what we have as a body system. You can read about that further, but the point that we're trying to make here is that um, from DNA to the human body, there's a whole lot of systems that work together to help our body function properly. So to summarize this, to better understand the whole, we dissect the system into pieces, and we study those pieces to better understand not only the system but also those parts. And that's basically what we do in system biology. We just did that with the human body. In genetics, we study how genetic informations are passed from parents to offspring and the impact on variation of those genetic information. And so um, genetic analysis basically contributes to system biology. And so that's how these things are connected. And so our DNA is a four-letter alphabet. Uh, we're not going to go too deep into it, but the main takeaway here is that our DNA is a four-letter alphabet, which includes A, C, C, and G. And basically, this C stands for cytosine, G stands for guanine, A stands for adenine, C stands for thymine. And we talked about the fact that the collection, the sequence, genes are sequences of these, we call them nucleotides. So I'm going to say nucleotides. Um, so our whole DNA sequence, uh, all of it is called genome, like I mentioned earlier. It carries information about our being, which includes information about our height, our color, and how we may react to certain medication, and even our predisposition to certain disease. Those information are stored and transmitted thanks to the arrangement of these bases in a double helix, a double-stranded helix. As you can see here, this is what we call double-stranded helix. And basically, the fact that they're organized in double-stranded helix is to allow for all the information to be captured inside the cell. If you think about the size of the cell, it's very microscopic, right? And you think about the fact that all those information is not just inside the cell, it's in the nucleus of the cell, which is even smaller. And if you look at the sequence, if you were to measure our whole genome, it will be more than 2 meters. It would be about or a little over 2 meters, 2 meters. And so you can think about how all these information at a microscopic level and with such length can fit in the cell. And it's because of these arrangement. It's because of these arrangement and uh, arrangement beyond the double helix that we're not going to go into. It's called chromatin. And so for that information, for such a long information to fit into the nucleus of the cell, it has to be arranged in a certain way. And so the DNA exists in a what we call double-stranded helix. And the bases are linked together, are linked across the two strands in a specific manner using hydrogen bonds. So C can only pair to G. Cytosine can only pair with guanine. And then adenine can only pair with thymine. And this is the way it works. That's the only way they pair together. So, and it's the order of these bases that form what we call the genetic code. And that genetic code sequence form what we call genes, which have boundaries, a start codon and a stop codon. And what we call codons here are words that we use to read the DNA, how the DNA is read. So the DNA is read in word of 3 letter long called codon. A codon is a code for a specific amino acid. So if you take three random nucleotides, it forms a codon. And amino acids are an alphabet of 20 letters. And basically, this is a table that summarizes how you go from uh, DNA letters, DNA letters to amino acids. And so in the table, you can see that there's no timing. The T, we just said that the DNA is a four-letter alphabet, which includes A, C, what's going on? Yeah, A, T, C, and G. And in the table here, you see that the T is missing. That's because often the often to translate into amino acid, the DNA will be converted into RNA, and the RNA only has what we call the uracil instead of the thymine. And you'll see, or you'll practice a little bit later how to convert these. I'll provide you some information about that. And so, in general, our genetic code shows that we have more in common than you might think. Genetically, we are all 99.9% similar. What that means is that no matter your height, your color, the color of your eyes, or where you live, we are basically genetically very, very, very similar. You can think about anything else and look at how similar they are. 99.9% seems like a great number, right? But while we're very similar, we're also unique. Our DNA shows that we are also we can also be very different. And that is very clear. Physically, you can see the difference among ourselves, even among twins, true twins. And you can look at your siblings, you can look at your parents, you can see that even though there are very big similarity, there are also some differences that may not be so subtle. And so the uniqueness, but at a molecular level, as you can see, we're very similar, but the way we find out how different we might be. You've heard about forensic analysis. If you look at the TV shows with investigation and others, they use a technique called DNA fingerprinting, which helps identify the differences among between two people, let's say that. And so um, DNA fingerprinting uses basically target some location in the genome that are highly variable, and they're called microsatellites. We're not going to go too deep into it, but the point that we're trying to make here is that genetically we're very, very, very similar, and we're also very unique as people. And so um, if you take away anything from this presentation, it's the fact that our genetic code is very universal, from human to animal to plant. Our genetic code is very unique. Uh, sorry, our genetic code is very universal, and so each amino acid is coded by more than one codon, as you saw in the table, and as you'll see a little bit in a few minutes. An example is the amino acid called arginine, which is coded by this codon AGA and also AGG. So translation and transcription are the process of converting DNA into RNA and from RNA to protein, as you see in this figure. And I'm going to provide this link later on Slack so you can check it out. And basically, this website helps you convert a sequence of DNA into RNA, and from RNA to what we call uh, polypeptide, which is a series of linked amino acids. Uh, and so, if you think about what we all or what we talked about uh, so far, you have this sequence, right? Uh, we said that DNA is often translated into RNA by replacing the timing with uracil. And for a given position in a codon, if any letter leads to the same amino acid, in this example, you have the example of the serine where the third base or nucleotide can be U, C, A, or G. The first two are the same, but the third it can be any nucleotide. So we usually replace it with N to say that it's any nucleotide. So if you see UCN, it's the amino acid called serine. So now I want you to look at this, to consider this DNA sequence. And I'm going to give you a few minutes to convert it into an amino acid sequence using the table here. You have a few minutes. I'm going to give you maybe 2 minutes. Um, Once you're done, you can post, you can drop your amino acid sequence in the chat. This is a pointer for you to see. Oh, wow. I think that's a so answer. Anybody has an answer?
I'm sorry, what was the question?
Uh, can you find the amino acid of the amino acid sequence of this DNA sequence using the table? I highlighted the codon here uh, as a visual help.
It will be GGGA, and that is glycerol. Your one highlighted will be GGA, and you've got your G in your last column under G, you've got like double GA. And you've also got a triple G. Must we only do the highlighted one?
Here. Jenny, you have a question? I think that
I'm so sorry, my mic cut off when you were speaking, so I'm a little lost of what you were asking us to do.
okay. So I told you guys earlier that DNA is often transcribed into RNA, and that transcription happens when the thymine becomes the uracil, and then you have an RNA sequence instead of the DNA sequence. So ATG becomes AUG, ATC becomes AUC. So any thymine becomes uracil, right? And DNA is codons are words of three letter are sequences of three nucleotides. And in this case, if you you divide this sequence into words, you have AUG, which is a word. You have AUC, which is another word. UCG is another word, right? Basically, by transforming the RNA into into amino acid, you're doing what we call translation. And so polypeptide is a series of amino acids that are linked together, linked together. And as an exercise, I gave you this table, and I want you to look at this sequence and translate it into amino acids using this table. So bear in mind that this will be translated into RNA, and for that you're going to replace every T with U.
Okay, thank you so much. That clarifies a little bit.
Yeah. And so each three letters is going to be a codon, and each codon is an amino acid that you have to find from this table. And to help you, I have highlighted the codons here.